Hey there, science fans! Welcome to another thrilling episode where we explore the wonders of the natural world. Today, we're diving into the exciting world of DNA, the blueprint of life. This tiny molecule holds the key to understanding who we are. It's the instruction manual that makes you, and me, every single trait we have is encoded in this remarkable molecule. From the color of your eyes to your height, DNA holds all the secrets. It even influences your susceptibility to certain diseases. DNA stands for deoxyribonucleic acid. It's a term you'll hear often in genetics and biology. It's a long, complex molecule found in the nucleus of almost every cell in your body. Imagine it as a tightly coiled thread packed with information. Think of DNA as a giant library filled with books. Each book contains vital instructions. Each book is a gene, holding instructions for building and running your body. These genes are the chapters of your biological story. Genes determine traits like hair color and blood type. Pretty cool, right? They even play a role in your personality and behavior. DNA is essential for life. Without it, cells wouldn't know how to function or replicate. It carries genetic information from one generation to the next, ensuring the continuation of species. This transfer of information is fundamental to evolution. Understanding DNA and how it works is crucial for advancements in medicine, agriculture, and many other fields. It helps us fight diseases and improve crops. So buckle up, science enthusiasts. We're on a journey to uncover the secrets of life itself. We're about to unlock the secrets of DNA replication, transcription, and translation, the processes that make life possible. These are the mechanisms that ensure our cells function correctly. Get ready to be amazed by the wonders of molecular biology. The more we learn, the more we realize how incredible life truly is. Picture a ladder twisted into a spiral. That's DNA. This twisted ladder shape is called a double helix. The sides of the ladder are made of sugar and phosphate molecules. The rungs are made of special chemicals called bases. There are 4 different bases in DNA: adenine, A, thymine, T, guanine, G, and cytosine, C. These bases pair up in a specific way. A always pairs with T, and G always pairs with C. These base pairs are like the letters of the genetic alphabet, carrying the instructions for life. The sequence of these bases determines the genetic code. It's like a language that cells can understand. Each 3-letter sequence, called a codon, codes for a specific amino acid. Amino acids are the building blocks of proteins, which do most of the work in our cells. Understanding the structure of DNA is key to understanding how genetic information is stored, replicated, and passed on to future generations. It's the foundation of molecular biology. Imagine you have a very important instruction manual, and you need to make a copy. This manual contains all the essential instructions for building something crucial. That's what DNA replication is all about. It's like making a precise copy of an instruction manual. Every time a cell divides, it needs to make a copy of its DNA for the new cell. This is essential for growth, repair, and reproduction. This process ensures that each new cell gets a complete set of instructions. Without it, cells wouldn't function properly. First, the DNA molecule unwinds and separates like a zipper unzipping. This is the initial step in the replication process. Each strand of the DNA ladder acts as a template for a new strand. This ensures that the new DNA is an exact copy of the original. Special enzymes called DNA polymerases help match up free-floating bases to each template strand. These enzymes are crucial for the accuracy of the process. A with T and C with G. They always pair up correctly. This specific pairing is what makes the replication process so reliable. As the bases pair up, 2 new DNA molecules are formed, each identical to the original. This duplication is fundamental to life. It's like magic, but it's science. The precision and efficiency of this process are truly remarkable. Now each new cell has its own copy of the genetic instructions. This is vital for the cell's function and survival. DNA replication is a complex and highly accurate process. It involves numerous steps and components working in harmony. It ensures that genetic information is passed on faithfully from one generation of cells to the next, making life possible. This continuity is the essence of biological inheritance. Let's say you want to bake a cake. You have a cookbook with all your favorite recipes. You wouldn't carry the whole cookbook to the kitchen, right? Instead, you just copy the specific recipe you need onto a piece of paper. You just copy the recipe you need. This way, you have only the necessary information at hand. That's what happens in transcription. It's like copying a recipe from a cookbook. DNA holds all the genetic information, but it stays safe inside the nucleus, much like your cookbook stays on the shelf. RNA, a single-stranded molecule, is like the messenger that carries the instructions to the protein-making factories of the cell, the ribosomes. It's like taking the recipe to the kitchen. In transcription, an enzyme called RNA polymerase binds to a specific region of the DNA molecule, much like finding the right page in your cookbook. This region contains the gene that needs to be transcribed, similar to the recipe you want to copy. The DNA unwinds and RNA polymerase reads the DNA sequence, creating a complementary RNA molecule, just like copying the recipe onto paper. The RNA molecule, called messenger RNA, mRNA, carries the genetic code from the DNA in the nucleus to the ribosomes in the cytoplasm, like taking your recipe to the kitchen counter. Think of mRNA as a recipe card carrying instructions from the cookbook, DNA, to the chef, ribosome. The chef then follows these instructions to make a delicious cake. Transcription is a crucial step in gene expression, allowing cells to use the genetic information stored in DNA to create the proteins they need to function. Just like following a recipe is essential to baking a cake. Section 5. Translation, turning RNA instructions into protein powerhouses. Finally, we're ready to bake our cake. In translation, the mRNA molecule carrying the genetic code arrives at the ribosome. The ribosome acts like a chef, reading the mRNA instructions and assembling the amino acids in the correct order to make a protein. Each three-letter codon on the mRNA codes for a specific amino acid. The ribosome reads the codons one by one and transfer RNA, tRNA molecules, bring the corresponding amino acids to the ribosome. Like a well-coordinated dance, the amino acids are linked together in a chain, following the mRNA instructions. This chain of amino acids folds into a specific three-dimensional shape, forming a functional protein. Proteins are the workhorses of the cell, carrying out a wide variety of tasks, from transporting molecules to catalyzing reactions. Translation is the final step in gene expression, turning the genetic information stored in DNA into the proteins that make life possible. Section 6. The central dogma, DNA to RNA to protein, a fundamental process of life. We've just explored the amazing journey of genetic information, from DNA to RNA to protein. This flow of information is known as the central dogma of molecular biology. It's a fundamental principle that governs how life works at the molecular level. DNA replication ensures that every cell gets a complete set of genetic instructions. Transcription copies those instructions into mRNA molecules, which act as messengers. And translation uses those mRNA instructions to build proteins, the workhorses of the cell. This intricate process is happening right now in every cell of your body, making sure you have all the proteins you need to function. From digesting your food to thinking your thoughts, it all starts with the central dogma. Understanding the central dogma is essential for understanding how life works at the most basic level. It's a cornerstone of modern biology. Section 7. The importance of getting it right. Errors in replication, transcription, and translation. Like any important process, DNA replication, transcription, and translation are not immune to errors. Sometimes mistakes can happen, leading to changes in the DNA sequence or the production of faulty proteins. These errors can have a wide range of consequences, from harmless to harmful. Mutations, changes in the DNA sequence, can be caused by errors in replication or exposure to mutagens like radiation. Some mutations are silent, having no effect on the organism. Others can lead to changes in protein function, which can be beneficial, harmful, or neutral, depending on the specific mutation. Errors in transcription and translation can also occur, leading to the production of non-functional proteins or proteins with altered functions. These errors can disrupt cellular processes and lead to various diseases. Understanding the causes and consequences of errors in these processes is crucial for developing treatments for genetic disorders and understanding the evolution of life on Earth. Section 8. Beyond the Basics. Exploring the vast world of molecular biology. We've just scratched the surface of the fascinating world of molecular biology. There's so much more to explore, from the intricacies of gene regulation to the cutting-edge techniques used to study DNA and proteins. Scientists are constantly making new discoveries about how DNA works, leading to advancements in medicine, agriculture, and biotechnology. For example, Gene editing technologies like CRISPR-Cas9 are revolutionizing our ability to treat genetic diseases and engineer crops with desirable traits. The study of molecular biology is essential for addressing some of the world's most pressing challenges, such as developing new treatments for diseases like cancer and Alzheimer's, combating climate change, and ensuring food security for a growing population. Section 9. Keep exploring, science fans. So there you have it, a glimpse into the amazing world of DNA replication, transcription, and translation. From the structure of DNA to the central dogma of molecular biology, we've covered a lot of ground today, but this is just the beginning. There's a whole universe of knowledge waiting to be discovered in the field of molecular biology. So keep asking questions, keep exploring, and keep that love of science burning bright. Who knows what amazing discoveries you might make? Remember, science is everywhere, from the smallest cell to the largest galaxy. Keep your curiosity alive, and never stop exploring the wonders of the natural world.